Hello, my name is Dina Svetlitsky. I'm a PhD student at Ben-Gurion University of the Negev, and I'm going to present to you an algorithm and a tool for the discovery of multi-operon collinear syntenic blocks in microbial genomes. This is a joint work with Tal Dagan and Michal Ziv-Yukelson. Microbial genomes are usually small, with a few thousand genes. They evolve rapidly by mutations, rearrangements, and also by transfer of DNA. Microbial genomes are typically organized in operons. These are co-regulated genes that are co-transcribed and co-translated. Despite extensive shuffling of microbial genomes, functionally related genes may be constrained to remain close to each other due to natural selection. We use the term syntenic block to describe a group of genes that are located close to each other in multiple genomes. A syntenic block is more general than an operon, and it can represent an operon, a suboperon, multiple operons, or in general a group of genes that are conserved together. An important computational problem is to discover syntenic blocks in a given set of microbial genomes using comparative analysis. Why are we interested in the discovery of syntenic blocks? The functional annotation of many microbial genes is still unknown. Therefore, the identification of syntenic blocks can help discover new biological systems. Here are two examples. One is the discovery of new bacterial defense mechanisms that are conserved as syntenic blocks. And another example where syntenic blocks helped recovered functions that are specific to the interactions between plant-associated bacteria and their hosts. Some previous analysis suggested that the order of genes in an operon or in a syntenic block is often conserved. Some explanations for gene order conservation is that gene order is optimized for ordered protein complex assembly or for the performance of consecutive reactions in a metabolic pathway. Therefore, conservation of gene order can be used for the prediction of functional associations between genes. In our research, we focus on the discovery of collinear syntenic blocks, in short CSBs. These are syntenic blocks where the gene order is conserved among all instances of the pattern. This is a pattern discovery problem where the pattern is not known in advance. Formally, we are given as input a set of genomes that are represented as strings over an alphabet of gene orthology groups and user-specified parameters Q and K. A CSB is characterized as a pattern and its instances. The pattern is a sequence of genes that abides by the following requirements. First, it must appear as a substring in one or more of the input genomes. And second, it is required to have instances in at least Q genomes, where each instance may differ from the pattern by up to K gene insertions. In this figure, the gene insertions are marked in red. They represent intruding genes that are not part of the pattern. The assembled microbial genomes available in public databases are currently in the range of tens of thousands and still growing rapidly. This opens up new possibilities for large-scale comparative analysis. However, previous tools for syntenic block discovery do not scale up to handle thousands of genomes. In a recent work, we proposed an algorithm for CSB discovery that does scale up to thousands of genomes. However, it was optimized for the discovery of CSBs that are confined to a single DNA strand. Single-strand CSBs usually represent a single operon or a suboperon. In that work, we applied the suffix tree-based algorithm to solve the problem. In the current work, we extend the problem to handle multi-strand CSBs that can span one or more consecutive operons. These CSBs are usually longer and could have a larger number of gene insertions. To accommodate for this, we design an algorithm with a time complexity that does not depend on the parameter k, and indeed is faster in practice for larger values of k. Before I go into the details of the proposed algorithm, I will show you one example of a multi-operon CSB that can be retrieved using the algorithm. Here you can see a CSB that contains genes encoding proteins of the bacterial flagellum, which is a tail-like protein complex that helps the bacteria move through liquid and track down its food supply. And here we see some of its instances in several bacterial genomes. The CSB consists of two operons that are collinearly conserved on different strands. As you can see, some instances have a large number of gene insertions. These gene insertions are highlighted in red. In this example, some of the gene insertions encode for proteins that are involved in the chemotaxis process, which is the movement of the bacterium towards a chemical or away from it by controlling the movement of the flagellum. In this work, we propose an algorithm for multi-operon CSB discovery. We incorporate the algorithm in a tool called CSB Finder S. I will now go over the workflow of the tool in high level. The tool takes as input a set of genomes. Each genome is represented as a sequence of genes, where the genes are annotated by their assignment to their corresponding gene orthology groups. In our analysis, we used COGs as gene identifiers. COGs are manually created gene orthology groups. In the first step of the pipeline, CSBs are identified in the input genomes. For CSB discovery, we use a sparse dynamic programming algorithm based on matchpoint arithmetic, which I will describe later in the presentation. In the second step of the pipeline, each of the discovered CSBs is assigned a probabilistic ranking score. Assuming uniform random order of genes in a genome, the ranking score of a CSB reflects the probability that it occurs by chance. This score is affected by the length of the CSB, the number of genomes in which it appears, the number of gene insertions allowed, and the frequency of the genes in a CSB. In addition, the score computation takes into account parameter delta that allows the user to adjust the score of the CSB according to the genomic distances between all genomes in which the corresponding CSB appears. This way, CSBs found across distantly related genomes will be ranked higher. In the last step of the pipeline, the CSBs are clustered to families based on gene content. So CSBs that have similar genes, or CSBs with the same genes and different gene orders, will be assigned to the same family. Now, let's go over the main points of the algorithm using an example. The input to the problem is a set of M strings of average length n over an alphabet of gene orthology groups. Here in the example, we have three input strings, S1, S2, and S3. By definition, the pattern for a potential CSB is a substring of one of the input strings. We don't know the pattern in advance, so we have to use a sliding window that goes over all input strings. We also don't know the size of the window in advance, so we will try different window sizes. Naively, there are O of n squared m squared candidate patterns to be considered, and for each candidate, we would need to compute all its instances. However, this computation can be done incrementally in an efficient way by utilizing some properties of the model and the data. To illustrate the computation, consider a table where the rows represent the input string S1 and the columns represent the other input strings. The cells in the table that are marked with the letter X correspond to match points, where the letters in the two compared strings are the same. For example, the first character in S1 is a B, and the second character in S2 is also a B, so this cell is marked as a match point. The algorithm considers all start indices from S1, and for each start index, it incrementally extends the considered pattern. Now, let's start by considering a pattern consisting of the first character of S1. As the pattern is extended, the algorithm forms chains of match points, which represent the instances of the pattern. A pattern that appears in at least q input strings is reported as a discovered CSB. When appending a match point to a chain, each skipped column corresponds to a gene insertion. In this example, we see the insertion of the character D in the instance represented by the rightmost chain. Our algorithm achieves its efficiency based on a combination of the following techniques. First, the number of match points is small with respect to the size of the table, and the proposed algorithm employs an efficient chaining of the sparse set of match points. Second, the instances are represented by a compressed set of chains consisting of the canonical representatives with the smallest number of insertions. And third, the search space is pruned by the parameters k and q. In this example, k equals 2. Therefore, the last chain is not extended, as it will result in 3 character insertions. In addition, in this example, the quorum parameter is 3. So, if the pattern BAC appears in less than 3 input strings, the pattern will not be extended. This is correct because as the pattern is extended, the number of its instances can only decrease or stays the same. The proposed algorithm exploits these properties and therefore is very efficient. The running time complexity of our algorithm depends on the OCC parameter, which denotes the number of match points participating in all the reported instances of the discovered CSBs. In our data analysis, OCC is roughly linear in the input size. The algorithm and the workflow described earlier was incorporated in a tool called CSB Finder S that has a graphical user interface that facilitates selective navigation through the results. The algorithm was implemented in a multi-threaded mode, which further contributes to its scalability. The dataset that we used for the analysis consists of about 1,500 microbial genomes, spanning 31 phyla. Running the tool on this dataset took about 40 minutes on the machine mentioned here, but is also practical to run it on a standard laptop. How frequent are collinear Syntenic blocks? We harness CSB Finder S to re-examine whether Syntenic blocks are indeed collinearly conserved. Our results show that about 76% of the gene sets in our dataset appear exclusively in only one gene order. For this analysis, we considered all sets of genes that appear in our dataset consecutively without gene insertions, and we considered only gene sets that appear in at least 30 genomes. A gene set consisting of 2 genes, for example, can have up to 4 different variants of strand and gene order combinations. Now let's look at gene pairs. The dark gray part of the chart corresponds to gene pairs with 2 or more gene order variants, while the light gray part corresponds to gene pairs with only 1 gene order variant. If we look at gene pairs, it seems that about half of them are not conserved in a particular gene order. However, although larger gene sets have a greater number of optional strand and gene order variants, larger gene sets tend to be more exclusively collinearly conserved than the smaller gene sets. For example, 97% of gene sets of size 7 are exclusively ordered. Note that roughly half of the gene pairs are subunits in longer conserved gene neighborhoods. You can think of it as a Russian doll concept. In this example, the most common variant of the gene pair AB is part of a longer conserved gene context consisting of 4 genes. In the next slide, we will show how this concept can be used by biologists for exploratory analysis by examining the extended neighborhood context of a gene pair of interest. Biologists are often interested in specific genes and their genomic neighborhood. For example, consider the gene pair pool E pool F, which is a highly conserved gene pair. This gene pair has 2 gene order variants, where one of them is very common. Using our tool, we observe that only the common order variant pool EF appears as a substring of longer CSBs, while the rare order variant does not. Now let's exemplify how different functional contexts of the gene pair pool EF can be studied using our tool. Using the filtration options provided by our tool, we extract CSBs containing both genes. Here are some of the CSBs that were extracted along with their taxonomic distributions, which are also reported by our tool. The first 2 CSBs contain genes from the type 2 secretion system, which is a system that allows the bacteria to secrete a variety of different proteins, including bacterial toxins. In CSBs 3 to 5, The poly-F gene pair is present in the context of DNA uptake machinery that allows the bacterium to take up extracellular DNA from its environment. The other two remaining CSBs contain genes that are part of the type 4 pilus system. The pili are fibers formed at the surface of many bacteria and have many functions, such as adhesion to host cells, twitching motility, and DNA uptake. Genes that function in these three contexts share several structural and functional features and were previously shown to be evolutionarily related. We also observed that three of the CSBs across two distinct systems, DNA uptake and pilus assembly, contain genes involved in resistance to antimicrobial substances. This is not surprising, as these systems are involved in the virulence pathways of pathogenic bacteria. In this example, we illustrated several multi-operon contexts surrounding the poly-F gene pair. To enable biologists to search the discovered CSBs based on context, we provide several filtration options in the user interface of CSB Finder S that support functional semantic-based queries. In conclusion, our aim was to discover cross-strand multi-operon CSBs in large microbial datasets. We presented an efficient algorithm based on sparse dynamic programming that is insensitive to the number of allowed insertions and scales up to thousands of microbial genomes. By analyzing a large dataset of microbial genomes, one of our results yields a strong assertion to the observation that gene order is highly conserved in Syntenic blocks. The CSBs uncovered by our tool likely represent functional units whose expression is tightly coordinated and their gene order evolves under strong selection. We invite you to download the tool from GitHub and use it for your analysis. Thank you.